excited to introduce our next presenter. So today we have Dr. Repka. She's an assistant professor of chemistry and biochemistry at Middlebury. And her lab focuses on developing new methods for identifying the biological targets of molecules that have potential to act as drugs because of their biological activity. So she's here to talk to us about some of the recent research she's been working on in her lab. And I'm excited to have you hear about the work she's been doing. So let's welcome Dr. Repka.
Hi, everyone. I hope you're doing well this afternoon. Thank you for being here. And thank you, Emily, for the very nice introduction. I appreciate the invitation to be here. So, yeah, as Emily said, I'm an assistant professor at Middlebury, and overall my lab is interested in new methods that can help with identifying the targets of drugs or potential drugs. And mostly at the moment, our research is focused in the area of organic chemistry. We also do some biochemistry. And we're going to get to this reaction, which is our goal at the moment. We'll see this significantly later on, but just this is a snapshot of where we're trying to go. We're trying to develop a photocatalytic reaction. That means it involves light and a photocatalyst, which we'll talk about more what a photocatalyst is. And we're trying to make a reaction happen between amino acids and alkoxyamides, which is what this group in blue is. It's an alkoxyamide. We'll talk about this in a lot more detail. But we're trying to make alkoxyamides and amino acids, this is a derivative of amino acid, react, and in that way helping us with target identification. But we'll get to that later. Overall, a lot of interest in photocatalysis and alkoxyamides. So I wanted to start with a little bit about my background. Going way back in time, about half a lifetime ago. I'm from Baltimore, Maryland. I went to the Friends School of Baltimore. I don't know if there are any Friends schools in Vermont, but there are quite a few of them in the Mid-Atlantic region. That's where I went to high school, and I had an amazing chemistry teacher. That's basically the reason I continued to pursue chemistry. And I ended up going to Barnard, which is a women's college in New York City. And when I was there, similarly, after the first 2 semesters of organic chemistry, that was what I wanted to do. I was really interested in organic. I had a great experience with it. And ultimately, I ended up pursuing research as an undergraduate. It was a couple of years into undergraduate. I didn't start that until the summer before my senior year. But I did research both at Barnard, doing organic chemistry research, as well as at Johns Hopkins. I worked at Johns Hopkins over the summer before senior year and then did a senior thesis at Barnard. And those experiences were really valuable for me. I loved being in the lab. Working at Johns Hopkins, which was partly so I could be at Baltimore and live at home over the summer, was a great way to see what graduate school might be like. And I really enjoyed that experience. And my senior year of college, my favorite thing was being in the lab. I loved it. So that was a pretty good indication to me that maybe going to graduate school might be something I would enjoy. My route is very linear, and I wouldn't, I meant to ask, can you hear me? Okay, awesome. All right, before I keep going any further, if you couldn't hear me, that would be a problem. This is a very linear trajectory, I think. I didn't necessarily think about all of the options I had, and I encourage my students to explore those options. There's a lot of possibilities for careers in science, journalism, consulting, law, in addition to academia and industry and many others that I'm not thinking to mention at the moment, which I didn't really consider. I just went straight through, but I encourage you to think about all these possibilities. But my trajectory was quite linear. Then I went to Caltech for graduate school, enjoyed a few years in Southern California, and did Ph.D. work in organic chemistry. And then my advisor, all of the advisors were incredibly supportive along the way, recommended that I try something a little bit different for my postdoc, which a postdoc is basically in most cases required if you want to go into a tenure track academic position. But it was also a really good opportunity, as advised by my graduate advisor, to get some experience in a completely different field. And so I went on and did my postdoc in biochemistry at the University of Illinois in Urbana-Champaign. And one of the things that that's allowed us to do is to, as I briefly mentioned, use organic chemistry and biochemistry together in my lab at Middlebury. And I like the variety, and it also helps us to do more with the research. So that's what led me to Middlebury. I didn't include it on the slide, but our research is in chemical biology, which is basically merging organic chemistry and biochemistry together and developing organic reactions that can help us to study biology is one way to define chemical biology. And this is about where we are now, half a lifetime later since high school. This is my lab in this past spring. I often put this slide at the end, but I wanted to put it first to give you a sense of my group and kind of where we are at the end of this journey, at least at the point where I am now. So I've worked with a lot of great students. We have really wonderful scientific support at Middlebury as well that has been critical. We also have some really nice instruments, which I've mentioned here, our JUL NMR and our Waters LCMS, which we'll get to later on. I'm going to show you how we use those. And then I just, while I'm on this slide, wanted to thank the Vermont Biomedical Research Network for funding this research, as well as Middlebury College. So this is us, by the way, on the yellow chair right in the middle of Middlebury. Have any of you been to Middlebury before? So you might recognize the yellow chair. Great, perfect place to take a photo. All right, so we're going to switch to talking about science at this point, but and I'll probably just keep talking, but if something's unclear to you and you want to ask, especially if it's going to make things confusing as we go along, I wouldn't want that. Please feel free to raise your hand or call out if I'm somehow missing your hand. I'm happy to address questions along the way too. Okay, so as I mentioned and Emily mentioned, we're interested in new methods that can help us identify targets of drugs, and it's really important to understand the targets of drugs. We wouldn't want to take a drug and not really know what it's going to do once it gets in our body. That wouldn't be safe, and it's precedented as being really unsafe. So thalidomide, which you might have heard of, does that sound familiar? Is a pretty well-known example of a drug that was not fully understood when it was first administered and led to very terrible effects. It was given to women who were pregnant because it helps with nausea, but it also has teratogenic effects which inhibit cell proliferation and led to death of fetuses, but also severe deformities if people were actually born. So it was really not good. This was in the 1950s and 60s. And today it's very difficult to have a drug approved if you don't have a pretty comprehensive understanding of its target and how it's functioning when it's in the body. This is thalidomide. It's kind of hard to say, sorry. And it exists as these two molecules, which are referred to as isomers, or more specifically enantiomers. One of them is an anti-nausea, has an anti-nausea function, but one of them has those teratogenetic properties. And they differ at this one atom in basically the spatial arrangement of atoms around that site. And I'm using an organic chemistry simplification at the moment that I'm going to need to use as we go along, but I want to make sure it's clear what this is that I'm showing. So I've also shown it like this. In organic chemistry, we don't draw molecules like that because that's a lot to take in. It's really hard to know where you're supposed to focus when you're seeing that many atoms at once. So basically, we've got all of the atom labels shown in that structure. Carbon is C, nitrogen is N, hydrogen is H, and O is oxygen. So the first letter of all of those atoms is their atom abbreviation label. When we draw molecules generally in organic chemistry, most of the carbons aren't shown. They're just illustrated by these little points where the line bends. We always show nitrogen and oxygen, but there's a lot of simplification that happens. Some hydrogens are shown, some are not. So, going back to thalidomide, for a really long time actually, it was not known what its targets were, and there has been renewed interest in this molecule thalidomide because of the fact that it's really not good if a fetus is growing to have cells not be able to proliferate, but if you're talking about treating cancer, you actually want to stop cells from proliferating. And so there has been use of thalidomide, repurposed use as a drug in treatment of certain cancers. And so recently there has been interest in particularly understanding a lot more, because it's going to be used as a drug, about how this molecule functions. And in 2010, about 50 years, yeah, go ahead.
I'm just wondering exactly what the molecules that you have up there, what does the dark line and the dashed line mean in like what's the difference between the nitrogen and the hydrogen bonding with the carbon at that site between like the dark and the dashed?
Thank you for asking. That's a very good question. The dark one, so this one, that darker wedge that's filled in, that means that nitrogen is pointing out of the screen at us. If there's a dashed line, that means it's going back into the screen behind us. And that's the only difference between these two isomers of thalidomide, is that for one, the nitrogen's going out of the screen, and for one, it's going into the screen. Thank you for asking. So we ultimately knew the target of thalidomide. One of them, at least, is a protein called cereblon. I think partly, maybe there wasn't as much research going on early on because people didn't realize that they could potentially use thalidomide for a good purpose. But target identification is also a really complicated and extensive process that I think also might be part of the reason why it took such a long time. About 4 years after people recognized that cereblon was one of the most important targets for thalidomide, there was also a crystal structure that was determined. You can grow crystals of proteins and then get these images of them. This is just a rough schematic of what the protein is. It's a simplification, and thalidomide is right here. You can see it binding, and that gives a lot more information about how thalidomide interacts with its targets, which is also really helpful, for example, if people wanted to come up with other drugs based on it. So as a bit of transition into the method that we are trying to develop for helping with target identification, I wanted to tell you how the target cereblon of thalidomide was determined originally. So here, as on the previous slide, I'm just showing a simplified drawing. I've left out most of the carbons. I also am assuming that both isomers might have been there, so I didn't show a wedge and a dash that time. But what they did was they attached thalidomide to a bead. That's a solid support. It basically looks like beads. And they did this by adding a linker, which is right there, to thalidomide so that they could connect it to the bead. So it's permanently tethered to the bead, or otherwise known as covalently tethered to the bead. And then they incubated this bead tethered thalidomide with cell lysate from HeLa cells. And so that cell lysate has a lot of different things in it. One of them is cereblon, the target of thalidomide, but there's all this other stuff, all these other macromolecules, many other proteins that are going to be around, carbohydrates, etc. There's no way to identify which is the target without getting rid of all of that other stuff, because otherwise it's just a sea of all of these molecules. So there's a lot of washing that happens at this stage, and that washes away everything that isn't the target of whatever molecule you have attached to the bead. And so presumably cereblon would then stick around if this process works well. And then at the end, what you can do is you can add in excess of the molecule that's attached to the bead, and that will bind really well to cereblon and displace. I didn't actually need to show this again, I'm sorry. We'll displace, I mean, it is in there, but what I meant to focus on is that cereblon, by addition of thalidomide at that stage, is now displaced. It's no longer binding to the bead because it's binding to thalidomide. And then can begin the process of actually characterizing the protein that you've isolated. But that's not the stage that we're focused on. We're focused on getting the target. But there would be a series of steps that happens beyond this to confirm that that molecule that's been isolated is indeed cereblon. This is often a really valuable, useful process. It works a lot. One of the challenges is that we can't really put beads inside cells. That's not exactly an option. And so we're not in a living cell, and that's going to potentially change the interactions that are happening. So we're changing the system that we're trying to study when we're not using living cells. In addition, there's a lot of washing that happens in this process, and we're going to do lots and lots of washing. If a target doesn't bind really tightly to a molecule, what might happen to the target? We're doing lots and lots of washing, keep washing. Yeah, it floats away. If this interaction isn't super strong, the cereblon in this example could be lost in all of that washing. So one way that people have come up with to prevent this challenge of all of this washing, making it potentially have your target get washed away too, is to permanently tether the target to the drug before all of those purification washing type steps, so that they're permanently tethered. No matter how much you wash, you shouldn't be able to separate them apart, and you'll still have the target at the end. A really common method that is used for this is called photocrosslinking. And what happens in photocrosslinking is that you'll have a molecule with some sort of handle on it, as it's referred to, and your molecule might be a drug or potential drug. The handle in this case is this red part. It's a diazurine. The handle is just the reactive part of the molecule. And what happens is that the molecule will be put into a living cell. Its target is in there somewhere too, amongst everything else. It's going to find its target in that living cell. And then what you do is you shine UV light on the cell. And we'll see a little bit about this mechanism, but the diazurine is going to undergo a change, and we're going to form a bond, a covalent bond, a permanent tether between the carbon that the handle was attached to and the target. I just want to show you a little bit about how mechanistically this happens. When you shine UV light on this handle, the diazurine, it releases dinitrogen. You can see there's this NN double bond here. You're going to form an NN triple bond and release dinitrogen, which is pretty favorable. You then get to what's called a carbene, where you have a lone pair of electrons, paired electrons on the carbon. That carbon does not have an octet. Anyone? What's an octet? Yeah. Ideally, atoms like to have 8 electrons around them. It doesn't currently have 8 electrons around them. Thank you for asking that. And because of that, it's pretty unhappy. it's going to keep reacting very readily because it wants a full octet of electrons. And it's going to react with the target to form the bond, that tether between them. Questions on that? We're starting to get into some mechanisms. I want to make sure that it's clear. Okay. All right, so we were here. We've got a molecule that has a diazurine. Briefly, if we imagine a similar process to that affinity purification before, except that now we're doing photocross-linking, we would have our molecule in a cell surrounded by lots of other molecules that we're ultimately going to have to get rid of. It's going to have that diazurine, that's its reactive group that allows it to undergo photocross-linking, but it's also going to have another organic handle called an alkyne. And we're going to shine UV light. So just bringing in here the same step as before, but adding the affinity purification that actually gets us to where we would be able to identify the target. So the molecule forms a tether to its target when you shine the UV light, but you still have all of that other stuff in there that you have to wash away. Sometimes people install biotin at this stage using that alkyne. That's another reaction. You might have seen biotin in other contexts. It's an affinity handle frequently used in biochemistry applications. But we still have all of these things around that we have to get rid of. But now we can purify, and that purification, I should have mentioned, involves, I should have written that there, involves a lot of washing. Again, like the other example that we saw with thalidomide, but in this case, we have a permanent tether between the molecule and the target, so they stick together throughout this whole process, and we have them isolated. Yeah.
So I'm wondering exactly how this process determines the affinity of the molecule to bind to the target. Like with the UV light going on there, I kind of, like there's not UV light going on in our cells, so how does this determine affinity?
Yeah, it determines affinity mainly because of the fact that the molecule and the target are going to be interacting before you shine the UV light. And we don't normally have UV light in our cells, and so for the most part, the only reaction that's going to happen is between that diazurine and the target. If other molecules underwent a lot of reactions similar to this using UV light, or if a lot of this functionality, these reactive handles were present naturally, this wouldn't work well. But it's because they're close together, and then when you shine the UV light, because they're next to each other, they form a bond.
So it's specifically the very controlled environment that gives you that information.
Yes. Yeah. But it's good that you brought that up right now, because UV light does not only do things to the molecule and its target. UV light's pretty detrimental. It's partly why we put on sunscreen when we go outside. It's not like UV light doesn't do anything. So for example, thymine, one of the bases of DNA, which is shown here with this schematic of DNA being shown, when you add UV light, you get a reaction that forms this square, this otherwise known as a cyclobutane, but it forms a square here. Additionally, UV light can be damaging to proteins. This is a protein that contains an SS bond, a disulfide, between S being sulfur. You have lots of disulfide bonds in your hair as well that are important for its structure. As well as in many other contexts, insulin has a disulfide. And what can happen with UV light is that disulfide gets broken. And that disulfide is really valuable for holding proteins, as roughly shown here, in a particular conformation. And if the disulfide's not there, if you break that bond that's holding it together, then basically your protein can unravel, which in other words means that when we shine UV light on cells, we can be having significant effects on the environment we study. If the target has a disulfide, that could make it difficult to actually characterize the target because your target might no longer be able to bind to the drug if it's no longer folded properly. So basically, to summarize, photocross-linking is an incredibly valuable method. It has been used extensively for target identification as well as many other applications. But UV light is one of its challenges, and really no method is without complications. So what we wanted to do was to hopefully complement this method using a reaction that instead of UV light uses visible light, which is less detrimental to our cells, but still can be used in living cells. Does that make sense so far? Okay. Any questions? All right. Okay, so I'm showing here, again, photocross-linking. The diazurine is one of the handles that's used for photocross-linking. There are a couple others, but it's a common one. With UV light in living cells, you can link a molecule containing a diazurine to its target. What we wanted to do instead, it was again use visible light and a photocatalyst. So we would have, in this case, a molecule that contains an alkoxyamide or an N-alkoxyamide. The N just means that the alkoxy group, which is this, is on nitrogen. But the molecule would have an alkoxyamide instead of a diazurine. And we anticipated that maybe using visible light, or specifically, we're using LED lights. Our hoods are often purple, although we cover them with aluminum foil to make sure that those lights are pretty bright. Purple and blue type lights are what we're using. And we hope that with a photocatalyst, we could achieve a similar reaction to photocross-linking, where a molecule via our handles linked to the target, but using slightly different conditions to do it. So I want to walk you through a little bit of mechanistically how we anticipated that this might be able to happen, because photocatalysis is a pretty different mechanism from photocross-linking. But overall, we're going to have a visible light, we're going to have a photocatalyst, which means it's a catalyst that's activated by light, that's the photo part. And the catalyst part means that it's regenerated at the end of a reaction, so it can keep being used over and over again. It remains intact, which is probably a good thing, not to have to add tons of photocatalysts to living cells when you're trying to do this reaction. We would need to add like much less because it's regenerated, at least theoretically. We'll have to wait and see what happens. So what happens in this reaction, mainly just to show the first step here, is that the photocatalyst reaches an excited state, which I'll talk a little bit about more what that is. It takes an electron off of the nitrogen of this alkoxyamide and generates a radical where we have, in other words, an unpaired electron on nitrogen and a positive charge. Similarly to what we saw with the carbene, nitrogen does not have an octet right now. It also has a positive charge. It doesn't really like that either. So it's going to react. And ultimately, we hope that through a series of steps, it will get to this product that we want. The mechanism actually is a little bit fuzzy once you get beyond the first step. There are a couple of different pathways that could be going on. So for the photocatalyst, just wanted you to see a little bit. I think it's pretty cool how these photocatalysts work and how they're then regenerated. I should preface this with electrons like to be in pairs, which is in some ways related to that octet rule, liking to have an octet. Nitrogen didn't want to have a single electron on it. Electrons like to be in pairs. I'm showing here really, as a really rough schematic of what electrons would look like, a pair of electrons indicated by these single-headed arrows. They're at a particular energy, indicated by the blue line. This other blue line is a higher energy. This is the ground state of a photocatalyst. It's at a low energy here. This is where it's going to hang out with its paired electrons. When you shine visible light, in our case generally purple light, one of those electrons, indicated by that single-headed arrow, is going to get excited, so to speak, into a higher energy level. And that's your excited state. And that's a pretty reactive state, being the excited state, because you have unpaired electrons. And those electrons don't want to be unpaired. They want to be paired with another electron. And so what's going to happen then is that this excited state is going to take that electron, as we saw up here, from the molecule, from that nitrogen, so that it now has created this pair of electrons. At that point, the photocatalyst is still in an excited state. It's not in its ground state. It's got, it's not in its original configuration, I guess. Maybe I should have, a different word than excited state is maybe what I should have used, but it's not in its original configuration where it can react again. And if you add in an oxidant of some kind at this point, it will remove this electron from that higher energy. Oxidants take electrons away and bring you back to the ground state, which can then do this reaction over and over. So we take something that has a pair of electrons, we make it into something that has unpaired electrons, and it really wants to have paired electrons, and that causes the photocatalyst to react with your substrate, with substrate being this, with your alkoxyamide, and make that reaction happen. That was a lot of mechanism. Any questions? You're still following? I wanted to introduce another term, which is this term called bioorthogonal. We talked about UV light and how it can be damaging to cells. In that sense, UV light isn't exactly bioorthogonal. What bioorthogonal means, or bioorthogonal reactivity means, is that some molecule can selectively react inside a cell without any of the rest of the biological system being affected by it. It remains inert. You don't want a reaction to happen with your molecule that you're trying to study and then have everything else in the cell start reacting too. That would be, you would be completely changing the environment you're studying. It would not work. So your reaction that you have has to be, so to speak, bioorthogonal, and nothing is perfectly bioorthogonal, but as close as you can get. Proteins are made up of amides, which look a lot like our alkoxyamide. An amide is defined by having a carbon double bonded to an oxygen. It's sharing 2 bonds with oxygen right next to a nitrogen. An alkoxyamide isn't present particularly in nature. It's not common, but amides themselves are. And so one thing that we needed to keep in mind in trying to develop this reaction is that we need an alkoxyamide to react, but we don't want amides, all the amides in your proteins, to start reacting. When the alkoxyamide interacts with the excited state photocatalyst and removes an electron, does resonance sound familiar? Does the term sound familiar to anyone? Basically, when electrons can move around a lot, they're happier. So if this single electron is stuck on nitrogen, it is not happy. By having this oxygen right next to it, that electron is free to move around some. So it's going to be a happier unpaired electron than if this oxygen weren't there. And those red arrows just indicate the electrons moving around. This shown in pink here is an amide. So similar to the alkoxyamide, but missing this oxygen here. The photocatalyst isn't going to be able to do a similar reaction, moving an electron from that nitrogen, because it's just that you don't have as much ability for the electrons to move around after the fact, which makes this a lot more unfavorable to happen. At least that's what should happen, and we're hoping that we will have something bioorthogonal in the end. We also thought we might be able to do photocatalysis in living cells because it's been done, not with this exact system, but people have started to use visible light and photocatalysis in cells, suggesting that we might not be damaging the system, hopefully not too much, and be able to study interactions between molecules and their targets within a living cell using visible light and a photocatalyst. All right, so I'm just showing again our approach using the N-alkoxyamide handle. We hoped we could use an LED light, a photocatalyst, and one day run this reaction in living cells and link these molecules and their targets together. We don't start with looking at a drug and a target and trying to link them together with visible light and a photocatalyst. That's a pretty complicated model system. We also don't know what the drug targets, so we wouldn't know if our reaction just weren't working well, or if the target were being degraded or something. There are a lot of variables once we start working in living cells with an actual drug and an actual target. And that's why we use a lot of organic chemistry. So this molecule, which could be a drug with an alkoxyamide, has been simplified to this. It's a much smaller molecule than a drug would be, but it still has that alkoxyamide. And what we hoped we would be able to do is react that with a variety of amino acids, forming covalent bonds to them, tethering the alkoxyamide to those amino acids, which would give you a structure like this. Those amino acids are a very small mimic of what a protein is. Proteins are made up of many amino acids, all linked together by these amide bonds. So this is basically the smallest mimic of a protein we could use to try to figure out if one day an alkoxyamide on the drug could be linked to a protein target. These aren't actually exactly amino acids. The part in red here is the amino acid part, although if it were actually an amino acid, we would have another hydrogen here and an H on that oxygen. It makes it so that we can use particular solvents. We don't have to run these reactions in water by not using exactly an amino acid. It's a so-called derivative of an amino acid. Where R, also shown here, is the side chains. So we've got these 20 amino acids and proteins, and they all have different side chains, but that red part is the same. So I'm just showing here again, this was the overall system that we were starting with. And this is the specific reaction that we started with. So the same alkoxyamide, but then using as our amino acid tyrosine, and using a photocatalyst, specifically eosin Y, and solvent being acetonitrile is what this is called, and our LED light, which is purple at about 427 nanometers. And this is the specific product that we hoped we would get there. But ideally, we would ultimately be able to use a wide variety of amino acids because of the fact that if you imagine a drug binding to a protein target, it could be near any of those 20 amino acids. And if we have a method that specifically only works for tyrosine, our method is not going to be helpful for target identification. We need the alkoxyamide to be able to tether to the protein regardless of where it is. I do want to point out that we started with tyrosine for a reason. This asterisk here, this purple asterisk, this is referred to as a benzylic carbon. This is benzene, which you may have heard about in other contexts as a pretty toxic substance. It's fine in tyrosine. The reason we started with this substrate is because there was a precedent. So we weren't doing this we had some precedent. It's not There are things that are new about it, but we wanted to make sure we had some precedent for what we were doing. And using a similar substrate, the same alkoxyamide a couple years ago, there were reactions that were done using this molecule, which is a derivative of benzene, but it has that same benzylic site, and it was undergoing reactions. So to form a bond between the benzylic site and the nitrogen of the amide, and it's a similar mechanism to what we were hoping to do. And basically, we don't necessarily anticipate that a bond will be formed to this site in our reaction, but we figured it might give us an extra site where a reaction could happen, increasing the likelihood that these molecules would form a bond with each other. And now we're going to get into some of the research that we've been doing. Questions that I can address? Is anything unclear that we should? Yeah, sorry.
So does this only work if they have the constant handle on it?
Does it only work if it has the handle on it? It should only work if it has the handle on it. Yeah, we should only be getting a reaction between these two molecules, or between this molecule and any amino acid, if that alkoxyamide is there. Does that answer your question?
We were discussing using like the photocatalysis and visible light on like living cells. What type of like research does that apply to, like in terms of like what kinds of cells would you be like wanting to test?
You might be using human cells like HeLa cells because they're so common. I haven't honestly thought too much about that stage yet, but you might be using something like that where you could maybe study a cancer drug. You might also be using a bacterial cell if you wanted to look at a drug that has antibiotic activity. So you might be doing that. Like Pseudomonas aeruginosa, which is a particularly dangerous pathogen, maybe you would be studying it in that. And you would get your molecule, you would incubate it with the cell. It has to be able to get into the cell, which is another important step. And then you would shine visible light when you feel like it's had enough time to get in the target.
And you would see how it processes with the cell, how it bonds.
Exactly.
Okay.
Yeah. And then you would go through a similar process to, as we went through with photocross-linking, you would pull out that tethered drug with its target, which really it's more than one target. There are off targets for drugs too, like that lead to side effects. So it would have many interactions with the cell, and you would purify those all out. And yeah, thank you. So I want to briefly show you the instruments that we use and give you a sense of the techniques, how the techniques work that we're using. These are some of my favorite instruments. I like spending time in these spaces. And they're really conveniently located to me. This is our NMR. It's a really nice NMR. This is basically a giant magnet. And NMR is nuclear magnetic resonance. MRI is the same concept, magnetic resonance imaging. It's just that in the medical field, they decided to avoid using the word nuclear. Nuclear here refers to nuclei, like of atoms. Not another kind of nuclear, but it's not called NMR in medicine, partly I think for that reason, just to avoid the confusion about what's going on. But it's very nice. Basically, we have an auto sampler, so we put samples in this holder here, and then this ramp takes them up and into here, into the magnet. They go down in here, and they're analyzed, and we'll talk a little bit about how that works. This is our LCMS. Which I'll come back to that, but it's liquid chromatography, mass spectrometry. just as a really brief introduction to NMR, I think you're still okay? Okay. Really brief introduction to NMR. What's going on when you run an NMR is that you have this really strong magnet that we just saw. That's a lot of that NMR instrument is its magnet. When we're looking at nuclei, and frequently that's hydrogen nuclei, so the center of these atoms, those atoms are also, they also have magnetic properties. We can think of them as little magnets. And they can be either aligned with the magnetic field, which is more favorable for them, or they can be opposing the direction of the magnetic field, and that's less favorable. They'd be at higher energy. What happens in an NMR is that we then apply energy, an energy pulse, it's a radio wave frequency, to the sample that happens inside the instrument. And some of those nuclei that were aligning with the magnetic field get excited to the higher energy state of opposing the magnetic field. And then ultimately they relax back down to their original state. How unfavorable it is to be opposing the magnetic field, or in other words, the energy difference between a nuclei aligning with and opposing the magnetic field, depends on how much electron density is around a particular nucleus. So just to show it schematically, if we have our little nuclei, and I've shown it here with only a few electrons around it, shown as E minus, there's a pretty large energy gap between those two states, aligning with and opposing the magnetic field. When there's a lot of electron density around a particular nuclei, then that gap is smaller. It's because electrons have their own little magnets, and those magnets tend to oppose the magnetic field, so it affects the relative strength of the magnet, and by extension affects that energy gap.
So this instrument measures electron density around these molecules, and measuring electron density gives you information as to how strong or weak that site on the molecule may be for something to bind to it.
The instrument is indirectly telling you the electron density around a particular nuclei because it knows what this energy gap is.
Okay, I'm just wondering specifically what value knowing electron density has.
That's a very good question. And I think hopefully this will help. If we look at our molecule, one of the main ones we've been focusing on, this alkoxyamide, it has two different sets of hydrogens. Hydrogens over here on the left, I'm ignoring this one here for the moment, hydrogens here on the right. They have different amounts of electron density around us. If we didn't know the structure of this molecule, the NMR would give us a sense of the extent of electron density around each of our atoms and would allow us to come up with what the structure is. So these set of hydrogens are really close to an oxygen atom. Oxygen's really electronegative. It likes to pull electron density toward itself. So that takes a lot of electron density away from these hydrogens, and the NMR that we'll see will reflect that. These hydrogens, on the other hand, are not as close to an electronegative atom, and so they have more electron density around them. They don't have something pulling as hard to remove electron density. Does that help?
Yes, and that information tells you how it would bind, right?
That information tells you how this molecule is bound together.
Okay.
Yeah. So I should have prefaced this. I don't know if you're still referring to the binding as in the reaction that we wanted to develop. Anytime we run those reactions, we can figure out whether or not our molecule and amino acid bound together by using NMR.
Okay.
Because of the variation, because we can tell the electron density around the nuclei. So I didn't quite make that transition, and I appreciate you bringing that up. This is what the NMR actually looks like for this alkoxyamide. There's solvent in there, a little bit of solvent, which is just, it's a reference. We need something to calibrate the data to. And anywhere where you have a hydrogen, you see a peak. And over on the left are hydrogens that are near less electron density. Over on the right are near high electron density. So hydrogens A, which are near more electron density, are over here. And further to the left, we have hydrogens B, which have less electron density. The other nice thing about NMR, besides telling us the electron density around atoms, which allows us to piece these structures together, is that it knows how many of a particular type of atom we have. We can take the area under these curves, or in other words, we can integrate them, and it will be representative of how many atoms correspond to that signal. So this integral is 3 because there are 3 hydrogens. And this peak is actually made up of 2 for hydrogens A, but they come out to about 3. So NMR tells us the number of the number of atoms we have, in this case hydrogens, in a particular environment, and it also gives us a sense of the electron density around them. Does that make sense? MRI is very similar, except that water molecules are what are generally analyzed, not something like this. All right. Unfortunately, when we ran NMR, which is basically the first thing you do when you run a reaction in organic chemistry, is you go take an NMR. We could not see, this was the first reaction we were trying to do, we could not see any of this by NMR, which was unfortunate because NMR is a quantitative technique, which I didn't mention. If the reaction were happening to any significant extent, we would be able to see it well by NMR. The fact that we don't see it means very little product is forming, very little of this. What we did see instead, and I'm not going to spend too much time here, I think we're a little low on time, was formation of, if we take this alkoxyamide by itself, we see formation of dimer. In other words, taking 2 of this molecule and binding them together, which does tell us that the first step of the mechanism is happening, which is great. This dimer is really only going to form if the excited photocatalyst removes an electron from that nitrogen. Otherwise, we can't get to that product. So step 1 is happening, but it's unclear why it wasn't happening beyond then. So we didn't want to give up. This is the dimer, but I think I might, for the sake of time, keep going and skip over this NMR, but similar analysis by NMR, we could tell dimer was forming. So what we wanted to do was look at this by LCMS, which was the other method that I mentioned before. This is the instrument. LCMS is a lot more sensitive than NMR. Basically what LCMS does is it tells you the mass of a molecule. And this particular LCMS tells you the mass of a molecule to 4 decimal places. And it's really unlikely to have multiple molecules that are the same mass to that many decimal places. So if you see a mass corresponding to the molecule that you think you've made that is accurate with those 4 decimal places, it's very likely that you have the right molecule. Just to summarize this method, what happens, the first stage, the LC, is liquid chromatography. The liquid part just being that the molecules you're trying to study, in this case, we hoped it was the product that we would form by an amino acid reacting with that alkoxyamide. The molecules are in liquid. They go through a column, usually made up of silica, the same kind of silica that you get in those, like, as a drying agent with silica packets that come in some mailings. This is the chromatography stage. It's basically packed lots of silica, a little more expensive than the little packets of it, but that the molecules travel through, and that allows them to separate out because they all interact with silica to different extents. Some of them have greater affinity for silica than others, and so some of them will have a hard time getting off the silica, and some of them will come off more easily. But what you end up with is a pure, ideally pure product. And this is the product we wanted to see. What happens in the next stage of mass spectrometry is that you have something called electrospray ionization, which basically just makes the molecules charged. It turns them into cations. You could add a hydrogen or you could add Na, which is sodium, but they need to be ions. In this case, cations. They need to be ions in order for us to be able to see what their masses are. And these are their respective masses. And then these ions, in the case of this instrument, go through this part up here, which I've shown very roughly here. This is the flight tube. This is a time of flight instrument. Heavier ions move slower. They fly slower than lighter ions. The instrument detects how fast a molecule is flying down the flight tube, and that tells it its mass. Because mass is directly related, there's an electric field that makes them fly, but the mass is directly related to the time of flight. Good? Does that make sense? So this is, again, the reaction that we wanted to see happen. These are the exact masses. This is what we needed to see by mass spec. And if we saw these masses with very little error by mass spectrometry, sorry, that's an abbreviation, by LCMS, we would be able to know if our product was there to some extent. And it is. This is a mass spectrum. These signals indicate ions, and they're at, on the x-axis here we have the mass. So this ion has a mass of 325.1396, which matches this, and then our other ion has a mass which matches the sodium bonded molecule. So, and we also did run some controls. I think I mentioned before, but I just wanted to mention again, this is related to tyrosine. This is a tyrosine derivative. If we don't have tyrosine, we don't see these masses. If we don't have the alkoxyamide, we don't see those masses. It's always good to have those controls because, as you can maybe kind of see here, the mass spec is an extremely sensitive method. That's why we can see these. Where we can't by NMR, it picks up on a lot. So these could have just been random artifacts, but they're not. And just really briefly, we also ran the same reaction with glycine instead of tyrosine, another amino acid, because as I mentioned, ideally this reaction that we're trying to do works with a lot of amino acids because otherwise it's not going to be helpful for target identification. A drug with this handle on it needs to be able to bind to a protein regardless of which amino acid it's by. And proteins are really common targets. I forgot to mention that. That's partly why we started with proteins. Other biological molecules are targets too, though. And similarly, the glycine reaction did work. But again, we don't see anything by NMR. So this is all very much a work in progress, and we've been trying different ways to improve the reaction. But this gave us some confidence that yes, the product that we want is forming. Maybe one day we could have applications in actually using this method for target identification. But we've got a ways to go before we're there. But I hope that one day we're there. I'm just going to summarize, I think, but questions on this before we get to the end? Okay, and then I hope I've saved time for questions. And again here, these masses matched our exact mass, which was in this case a sodium adduct. of the product that we were hoping to form matches what we see in the mass spec. So just to summarize, in target identification, one of the biggest challenges is that when you're trying to identify the target of a drug, if you use a method like that was used for thalidomide, sometimes you can accidentally wash your target away. And so it can be really helpful to permanently tether your drug molecule to its target so that that can't happen. And ideally, you can do this in living cells too, which that affinity pull down couldn't be done in living cells, and so you're changing the environment you're studying. And what we wanted to be able to do was to do a similar reaction, but instead using a visible light, or LED light in other words, for the reaction to happen, and using an alkoxyamide in place of something like a diazurine. But again, we wanted to link alkoxyamides to targets. And as I've just shown by LCMS, time of flight, we are forming the product. But the fact that we don't see it by NMR, I should have noted LCMS is a qualitative technique. Some things show up better than others. That doesn't mean there's a lot of them present. By NMR, we're not seeing anything, so we have some space to grow as far as getting to somewhat higher yield, and also looking at other amino acids that can react, and hopefully one day making it into a reaction in living cells. So that's all I have, and I appreciate you letting me know when you had questions throughout. That's helpful for making sure making sure that I'm being clear. And I would love to take as many questions as you'd like now that we have time for. Thank you.
Anybody have questions?
So why was it good to, sort of you're targeting these toxic chemicals in living cells? Is that the purpose of this experiment?
You might be targeting something toxic. I guess it would be probably most likely that your drug were targeting something that was toxic to the cell, and that was the mode of action for the drug, and you were trying to identify it. And you could use photocross-linking, or ideally one day our method to link whatever your drug is to that toxic molecule. You could also potentially, if you're just wanting to look at that toxic molecule itself and see what it's interacting with, you could add a handle to it and then shine light and see what it binds to. So you can look at something that's already in cells, or you can look at a drug that you're adding to cells. One way photocross-linking is also frequently used is to understand protein-protein interactions, which are frequently implicated in disease. And so you can have one protein that, that interaction can be toxic to the cell. You can modify one protein with the handle and then use this type of method to link it to its interacting protein and understand that interaction.
Okay. Thank you.
You're welcome.
Can you always put a handle on a drug, or there's some drugs that you just can't do that?
Yeah, that is one of the challenges. You're asking, just to make sure I'm following, about if you can always put a handle on the drug. Yeah. There are two reasons why it's sometimes challenging. One is that you have to have a site where you can install it easily. The nice thing with the alkoxyamides that we're trying to use is that you can incorporate them into a molecule if you have a carboxylic acid, which is the same thing as that alkoxyamide, except instead of a nitrogen, there's an OH, like in an amino acid. There's a carboxylic acid. So in that sense, they're pretty easy to install, and there are a lot of known drugs that have carboxylic acids, so that helps. Sometimes also installing a handle changes the activity of a drug. And so you also have to be really careful about which handle you choose, because no matter what, by adding a handle, we're changing the drug, and so we're potentially changing how it interacts with its environment. And where you install a handle could have a dramatic effect on the system you're trying to study. Does that answer your question? Thank you.
So thinking about just the way your lab is set up, and I know you have student researchers. So are the students kind of looking at your last sentence there? So are they working on individual sort of next steps, or are they working just on kind of certain aspects of the whole process?
It definitely varies. Most, and I mentioned their names on the slides, but I forgot to say them out loud. All of the data I just showed you was student data. Sometimes we're making substrates, sometimes we're making these molecules. So some molecules we can buy, they're commercially available. Some of them take a couple steps to get to. But they're also running the actual photocatalysis reactions. So it's really a mix of both. And at the moment, one of the things that we're really trying to do is think about, so we've switched this to a system where the molecule and the amino acid are, the alkoxyamide and the amino acid are part of the same molecule. They're linked together so that they're closer together at all times and they're more likely to react. We're relying on random collisions between molecules for this to happen. And some of us are looking at different substrates, different molecules, and thinking about which ones might have faster reactions when the alkoxyamide and the amino acid are part of the same molecule. A lot of different things. We're trying to figure out the best step forward at the moment. Any other questions? It doesn't have to be necessarily about target ID. If anything else came up that would be helpful to, or you'd like to learn more about.
So you had your PhD at Caltech. I addressed you as doctor, right? For all my paper. Okay.
Whatever, I guess. I go with what my students are comfortable with, but yeah, sure. Some of my family still writes it on things. It's nice. It's a little acknowledgement.
Yeah. Well, thank you so much.
I will see you guys. The plan is to be back in here next week. I'll send you any updates if anything changes, but so far.